When something freezes in the polar permafrost, almost everything that made it alive is supposed to just stop. The ice isn't just a physical barrier, it's a molecular tomb. When water freezes, it undergoes a transformation, expanding into razor-sharp crystalline lattices that slice through cellular membranes like glass. Inside a frozen cell, the machinery of life simply grinds to a halt. There is no metabolism, no replication, and no repair. Over tens of thousands of years, as cosmic radiation bombards the frozen soil and natural decay slowly eats away at chemical bonds. The delicate strands of DNA are supposed to break down into useless, shattered fragments. By all the laws of biology we've established over the last 2 centuries, the ancient Arctic should be a sterile graveyard. And that frozen dust, locked deep beneath the Siberian tundra, is supposed to be all we have left of the prehistoric world. Except it isn't, and a single microscopic entity revived in a French laboratory is the proof. The polar ice is the most hostile preserved environment on Earth, the one we thought we understood best of all. And we're still finding things deep inside it we can't explain. Things that show us we might be completely wrong about what's sleeping in the cold. That some of these ancient organisms might be totally different from anything we've ever seen. And that the ice might still have something locked inside it that shouldn't exist. It starts in 2014 with a block of 30,000-year-old mud and a plastic dish of live amoebas. And yes, that does sound like the exact setup to a bad, low-budget science fiction horror movie. But what happened next wasn't fiction. A team of French virologists, led by Jean-Michel Claverie and Chantal Bergel, decided to look into the vast, untouched expanses of the Siberian permafrost. They weren't looking for mammoths or ancient weapons. They were looking for something much smaller and potentially far more dangerous. They took a core sample of frozen earth from a depth of over 100 feet in the Kolyma lowland, a remote and desolate region in northeastern Siberia. This ground hadn't seen the light of day since the late Pleistocene epoch, a time when woolly mammoths and cave lions ruled the earth. Now the scientists faced a massive ethical and safety dilemma. If they simply thawed out the soil, they risked releasing a prehistoric pathogen that could trigger a global pandemic. To bypass this, they came up with a brilliant yet terrifyingly simple solution. They decided to use a process called amoeba baiting. They took a common single-celled organism called a Amoeba and placed it in a liquid dish. Then they dropped a small piece of the thawed Siberian soil into the center. The plan was simple. If there was an ancient virus trapped in that mud, and if it was still active, it would target the amoeba as its host. Because these giant viruses only infect amoebas, there was zero risk of a human outbreak. It was a perfect, safe experiment. But when they looked through the microscope, they saw something that shook their entire understanding of biology. What they saw under the light microscope was not a typical virus. When we think of viruses, we think of incredibly tiny, simple structures, microscopic packets of genetic material wrapped in a protein coat, so small they can only be seen with an electron microscope. But this thing was different. It was colossal. It was over 1.5 micrometers in length, making it easily visible under a standard low-power school microscope. It was actually larger than many species of modern bacteria. In the world of virology, this was the equivalent of finding a blue whale in a backyard swimming pool. Physically, it looked like an ancient Greek amphora, a massive, thick-walled oval jar with a strange cork-like structure capping one end. They decided to name it Pithovirus sibericum. But its size wasn't the most shocking part. The most shocking part was what it did. Within minutes of being exposed to the amoebas, this 30,000-year-old relic woke up. It didn't just drift aimlessly. It began to hunt. It attached itself to the membrane of the amoeba, slipped inside, and completely hijacked the host cell's internal machinery. It began to replicate, churning out thousands of copies of itself, until the amoeba's membrane stretched to its breaking point and burst open, releasing a fresh army of ancient predators into the dish. For the first time in 30,000 years, a pathogen from the Ice Age was alive, active, and highly infectious. It had survived the deep freeze, and it was still perfectly capable of killing. Well, nothing about this was supposed to be possible, so there had to be other explanations. In the scientific community, extraordinary claims require extraordinary proof, and the idea of a 30,000-year-old virus waking up was met with immediate, heavy skepticism. Many prominent virologists argued that the experiment was flawed. They believed that the fossilized soil sample had been contaminated in the lab by a modern giant virus that had somehow slipped into the equipment. It was a logical, comforting explanation. It was much easier to accept that a modern researcher had made a simple mistake than to accept that an infectious killer from the Pleistocene had survived frozen for nearly 300 centuries. The permafrost sample, after all, came from rock layers that had been firmly dated using radiocarbon techniques. There was no way a virus could survive that long without its DNA breaking down into molecular static. To settle the debate once and for all, Claverie and his team turned to genomic sequencing. They extracted the raw genetic material from the Pithovirus and began the painstakingly slow process of mapping out its entire DNA blueprint. If it was contamination, the genetic code would match one of the known modern giant viruses in their databases. But when the results came back, the skepticism turned into sheer disbelief. The genetic fingerprint didn't match anything on record. Instead, it revealed a massive, incredibly complex genome containing hundreds of genes. And, the truly terrifying part, a staggering 70 to 80% of those genes were completely unknown to modern science. This wasn't contamination. This was a biological ghost. It was carrying the genetic signature of a lost world, a blueprint from an ecosystem that had ceased to exist tens of thousands of years ago. Now, as I mentioned, this specific virus can only infect amoebas, which also means there's no threat of a global zombie plague from Pithovirus. What did actually survive the ice, though, might be equally insane. And now, let's get back to exactly what we've found locked deep inside the Siberian ice. When we look at how Pithovirus managed to survive 30,000 years of deep freeze, we have to look at what had to take place biologically for this to happen. Normally, when a cell freezes, the ice crystals physically rupture the cell membrane, spilling its contents and destroying its integrity. But giant viruses have evolved a completely different defensive strategy. Unlike modern, fragile viruses, like influenza or the common cold, these giant, prehistoric viruses are built like armored vaults. Their outer protein shell, the capsid, is incredibly thick and dense, composed of overlapping, highly resilient protein layers that act like a structural shield. This shield prevents ice crystals from penetrating the delicate genetic core of the virus. Furthermore, these viruses exist in a state known as cryptobiosis. In this state, all metabolic activity is completely suspended. There is no chemical reaction, no energy consumption, and no active cellular processes. The virus essentially behaves like a plant seed. It is completely inert, waiting in a biological pause menu for tens of thousands of years. As long as the environment remains consistently cold, dark, oxygen-free, and protected from light, the internal DNA remains perfectly preserved, cocooned inside its armored sarcophagus. But as we began to explore this phenomenon further, we quickly realized that 30,000 years wasn't even the limit, because for a long time we thought 30,000 years was the absolute biological ceiling for survival in the ice. But in 2022, the same French research team went back to Siberia to see if they could push that boundary even further, and what they brought back was even more shocking. They managed to isolate and revive 13 entirely new viruses from 7 different Siberian permafrost samples. These weren't just taken from soil, they were pulled from incredibly diverse ancient environments. They found one giant virus preserved inside the stomach of a frozen woolly mammoth. They found another locked inside the fossilized remains of an ancient Siberian wolf. But the most terrifying discovery came from a sample taken from a frozen lake in Yakutia. This was a sample of yedoma permafrost, an incredibly stable, water-rich, ice-cemented soil that has remained completely undisturbed for eons. When they thawed this sample and used their amoeba baiting technique, they woke up a giant virus they named Pandoravirus yedoma. When they ran the geological dating on the surrounding ice, they discovered it was over 48,500 years old. This virus was dormant when Neanderthals were still walking across Europe. It was frozen before the peak of the last ice age. And yet, after nearly 50 millennia of absolute silence, the moment it was thawed, it immediately began to infect and multiply. It proved that the biological storage capacity of the permafrost isn't measured in centuries or even millennia. It is measured in tens of thousands of years. And if a virus that old can wake up, it raises a deeply unsettling question. What else is waiting in that frozen waiting room? Up until recently, many critics argued that these discoveries were nothing to worry about. After all, these revived giant viruses only target amoebas, so they pose absolutely zero risk to human health or the global ecosystem. But in the summer of 2016, a terrifying real-world disaster proved that the dangers of thawing permafrost are not just confined to a laboratory dish. It happened in the Yamal Peninsula, a remote region of the Russian Arctic high above the Arctic Circle. This region is so cold that the permafrost stretches over 1,000 feet deep, acting as a natural permanent freezer. But in 2016, an unprecedented, extreme heat wave struck the Arctic, pushing temperatures up to a blistering 95 degrees Fahrenheit. This extreme heat began to melt the active layer of the permafrost, the top layer of soil that normally remains frozen year-round. As the ice turned to mud, it exposed a long-forgotten grave, the carcass of a reindeer that had died of anthrax more than 75 years earlier. The reindeer's body had been shallowly buried in the frozen ground, and for nearly 8 decades it lay perfectly preserved. But as the carcass thawed and warmed up, the dormant spores of Bacillus anthracis, the bacteria that causes anthrax, suddenly woke up. They didn't just survive, they thrived. The infectious spores spread rapidly across the tundra, carried by groundwater and blood-sucking insects. Within weeks, the outbreak ran rampant through the region, killing over 2,500 reindeer. But it didn't stop there. The disease jumped to the local nomadic Nenets herders. Dozens of people were hospitalized with severe infections, and a 12-year-old boy lost his life. It was a brutal, sobering wake-up call for the scientific community. The frozen ghosts of the past aren't just scientific curiosities. They are fully active, lethal pathogens, and they are starting to wake up. But the warming climate isn't even the most immediate threat we face. As the Arctic ice continues to melt at an accelerated rate, it is opening up previously inaccessible regions of the far north. Shipping lanes are clearing, and massive multinational corporations are moving in to exploit the vast, untapped natural resources buried beneath the ice. They are setting up massive operations for open-pit mining, deep-core gold drilling, and oil extraction. To build these facilities, workers have to physically excavate, blast, and drill through layers of permafrost that are not just tens of thousands of years old, but up to 1 million years old. These are layers of ice that were sealed long before Homo sapiens ever evolved. When these industrial operations dig up these ancient, undisturbed layers of Earth, they are exposing the workers directly to whatever prehistoric pathogens are trapped inside them. If a miner inhales a virus or bacterium that has been sealed in the ice for 500,000 years, they will be introducing a completely alien pathogen to the modern world. Our immune systems have absolutely zero evolutionary history with these organisms. We would have no natural antibodies, no vaccines, and no established medical treatments to combat them. It is a biological wild card unlike anything our species has ever had to face. And that is what makes the whole thing so deeply unsettling. We've spent the last century believing that the cold, frozen wastes of our planet were safe, static boundaries. We treated the Arctic as a pristine, beautiful tomb where the dangers of the ancient world were safely laid to rest forever. But the science is showing us that the cryosphere is not a graveyard. It is a waiting room. The giant viruses, the dormant bacteria, and the prehistoric pathogens are all still down there, locked inside the ice, waiting for the temperature to rise just enough to wake them up. So the next time you look at a photograph of a beautiful, pristine glacier or a vast, snow-covered tundra, don't just think about the quiet beauty of the ice. Think about what could still be hiding inside its bones, waiting for its chance to breathe again. And YouTube thinks you'll like this next video. If you want to support the channel, consider becoming a member. And as always,